focal point. Will it change if I move forward and backwards?
Seems to be auto-focusing. So it looked at what you see right there looks happy.
Okay. We good there? Okay, here we go. When we left off the last time we met, Wednesday, we had just gotten through the heartbeat. And I'm going to start there to get you up to speed. And then we're going to do the PQRST wave that you see on TV, on the monitors and stuff. Then we're going to go to blood, which is our next topic. And after blood, we'll do respiration. Okay, so here we go. When the ventricles contract, both, as we talked about before, both the right and left contract at the same time. That gives us our lub sound. We talked about that. As that happens, the blood is squeezed out of the ventricles and enters the pulmonary trunk going to the lungs on the right side, and the left side goes out through the aorta, both through semilunar valves. Okay? The pressure exerted on the ventricles, which we measure through the brachial artery, is called the systolic phase of the heartbeat. So systole or systolic is the high pressure we hear when we do the, we take up somebody's blood pressure, we hear the 120/80. This is the 120, the systole, the contraction phase of the heartbeat. When the heart relaxes, it doesn't go down to zero. There's still pressure because we talked about it being a pressurized system, and that's called the diastole. And that's what we usually hear as 80. So the 120/80 is the systolic over the diastolic. We call the diastolic the relaxation phase, and the systolic the contraction phase. Okay, now the next part we've already done. Remember, we drew a heart. And we drew our heart like this. And we put the 4 compartments, which are called chambers, like this. This being the right side, this being the left side. As you remember, the right side receives blood from the body into the right atrium. And then it goes through a valve called the right atrioventricular valve into the right ventricle. From the right ventricle, and I'll do it in blue because it's unoxygenated blood, takes it up to the lungs. In the lungs, it's oxygenated, and it comes back to the left atrium. Remember, we described the atria as receivers and the ventricle as pumps. Then it goes through the left atrioventricular valve, as you remember, that has 3 names. Left atrioventricular valve, bicuspid, and mitral valve into the left ventricle. From the left ventricle, it's distributed out to the body. The heart is autorhythmic. In other words, once and we're calling it electricity, but it's depolarization. Once that begins, it's autorhythmic. It'll continue through your whole life unless it's stopped for some reason. It begins with the sinoatrial node, which is located about right here in the right atrium. We'll abbreviate it SA node. That's also called the pacemaker. Here's where I am. Sinoatrial pacemaker. From the sinoatrial node, it goes to the AV node. AV node, called atrioventricular node. So here's what we've checked off so far. SA node, also called the pacemaker. AV node, also called the atrioventricular node. Using green, I'll show the electrical current. measured in millivolts. From there, it hops over to the septum, dividing the right and left side of the heart. And there's another node. Now remember, nodes are dendrites. This node is called bundle of His. So using a green arrow showing the pathway to here. Then the next, here's where I am, bundle of His. Now the interseptal fibers. Think of septal as septum. So this is interseptal fibers. That takes us all the way down to our apex. Remember the point of the heart, apex? From there, it sends depolarizations, we'll just use the word electricity, throughout the ventricles. And we talked about this through the intercalated disc is where the sodium travels. These are called Purkinje fibers. Anyway. The Purkinje fibers are where the ventricles contract, the cardiac tissue. So Purkinje fibers is right here. Remember, one, as you see, I drew one loop all the way back up here to the SA node, the pacemaker. And that's what makes it autorhythmic. And this continues 50, 60, 70 times a minute. Good? Okay, now can we measure this? That's the next thing. Can we measure the amount of electricity, which is measured in millivolts, through electrodes? And we do, and that's called the PQRST wave. And I'll draw it in a minute. Why they didn't do A, B, C, D, E, I don't know, but it's PQRST wave. And we talked about reading a graph. Remember when you see a picture of a graph, it's giving you the data, and the data is the answers to the questions. On the y-axis, we're going to put millivolts, and on the x-axis, we're going to put time. Most graphs do it just this way. Okay? Here is one cycle of PQRST wave. Now look what you're seeing. You're seeing a rise in the amount of electrical current going through this tissue at this time. And we can tell by where where these humps are, whereabouts we are in the heartbeat. The first rise is called the P wave. The second is called Q. Peak is R, S, and then the T wave. So cut it for a second, please. Again, we were talking about the PQRST wave and what was going on when and where. The small wave that you see, which is a measure of the depolarization, we say electricity, going through the heart at the atria. So P is atrial constriction. Remember we talked about that. We did the 80, 20%. Remember, 80% of the blood enters the ventricles from the atria because there's nothing in the atrium. The atrium, the ventricles are opening up. Blood is rushing from 100% in the atria down into the ventricles. When it goes down to the ventricles and it gets to 80%, the last 20% has to be squeezed in there, which means we can measure electricity, and that's the P wave. P wave is the measure of this 20, and that's why the hump isn't very big. Now, with the blood in the ventricles now, it has to be pumped to the lungs and to the rest of the body, which is going to take a lot of strength. That's why the measure of electricity through the heart is so dramatic. So, our our QRS wave is ventricular constriction. And remember, 100% is pumped. The T wave is repolarization of the ventricles. And I'll do it in black so you can see it. I said atrial constriction. It is also called depolarization. Ventricular constriction, depolarization. So once it's depolarized, now we have to repolarize it, and T is a measure of the repolarization of the ventricles. Now what's missing, you depolarize the atria, you depolarize the ventricles, you repolarize the ventricles. There's nothing that shows the repolarization of the atria because it's concealed here. So when this repolarizes, it's repolarizing in here and you can't see it. Okay, let's go over here to our checkoff list. We're talking about the PQRST wave. You remember the P is atrial depolarization. QRS is the ventricular depolarization, and now we know that the atria repolarize in there. And the T is the repolarization of the ventricles. We have a couple terms. Bradycardia. Now going back to your prefixes and suffixes we learned. Brady is what? Slow. Slow cardia. So that's a term when the heart rate has slowed to a certain amount of beats per minute. We give it the term you're in bradycardia. And that number is 60 beats per minute. 60. Your bradycardia. Tachycardia, tachy means quick, cardia is any time it's been determined to be 100 beats per minute. Now when you get into serious trouble, your heart will go into flutter. And that means it is beating so quick that the chambers can't fill back up with blood, and that's over 200, greater than 200 beats per minute. And then the last term I want you to know is fibrillation. Fibrillation is when there is no set pattern to the beat of the heart. In other words, it can constrict the atria. The way it's supposed to be is the atria constrict, the ventricles constrict. Atria constrict, ventricles constrict. When that pathway is off and the ventricles are pump, 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 then stop, slow down, pump, 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 that's called fibrillation. Okay, next topic. You're going to be asked on the exam about the PQRST wave. What is going on when? Now, next topic. As you remember, this was a 3-step lecture series. The first one was the heart, circulation through the heart, tissue of the heart, what the function of the heart was, chambers of the heart, stuff like that. Now, the second lecture was the blood vessels, the heartbeat itself. We did the descending aorta. We talk about branches of the descending aorta. We talked about capillaries. There's 3 capillaries grouped into 2 groups, continuous and discontinuous. In continuous, there was 1 type, continuous. In discontinuous, there was 2 types, fenestrated, small gaps, sinusoid, large gaps. And we talked about going from continuous capillaries to discontinuous capillaries. And we said that is triggered by an enzyme, a hormone enzyme called histamine that's released from the basophil. And that allows blood to escape the circulatory system and become interstitial. That process of the continuous capillaries becoming discontinuous is called diapedesis. Okay, now that brings us, so we did the vascular system. We talked about the 3 tunings. I'll bring it to you. I talked about this model in lecture, with the red being the descending aorta, the blue being the vena cava, the inferior vena cava. The inferior vena cava takes it up to the right atrium. Then you see the red arrow coming out of the left ventricle. This is part of that. It goes to the aortic arch and then down to the descending aorta. You notice that the descending aorta terminates into the iliac arteries. The vena cava begins with the iliac arteries. Okay. Now we're going to put blood into the vascular system. So we have the heart, which is the pump, the blood vessels, which carries it to the cells, and now we're going to take the nutrients and stuff, which is in the form of blood, to the cells. Reason we have that is the cells are static, they're stationary, they can't move. So what we're going to do is we're going to take their nutrients to them and take away the waste. And then once we get the waste, we're going to take it off to the kidneys to clean that up. So to begin with, what is the function of blood? Obviously, transport is one of it. It's transporting nutrients, transporting hormones, transporting everything to the individual cells. Next thing, regulation. Question in your mind should be, well, how does blood regulate anything? Well, just same way of what it transported. It's regulating hormones, it's regulating nutrients, it's regulating waste in the blood from the cells. Next one we see, protection. How does blood protect us? Okay, we know that our first line of defense is the integumentary system, skin. If we get a hole in that skin or a laceration, or better yet, in the circulatory system, I talked about is we have a closed circulatory system. Now we have an open system and we have to close it. The way we close it is using a blood clot. So we'll talk about the very end of the blood, we'll talk about a blood clot. Okay? So the next term we see, hemopoiesis. In order to have blood, you have to make blood. The term for making blood is hemopoiesis. Hemo, blood, poiesis, to make. And that there's 2 subcategories under that. The making of red blood cells, which is called erythropoiesis, and the making of white blood cells, called leukopoiesis. Good? And I'll ask you those terms on the exam. Now the components. I have a jar of blood. What is in it? Not just, so what is blood? Okay, I'm going to take you back to our first third of the semester when we were doing tissue. What is blood? Is blood a type of tissue? And your answer should be, yeah, it's connective tissue. You remember that? Okay, connective tissue has 2 things in common: cells and matrix. Remember that? Okay, the matrix has 2 components: ground substance and proteins. Okay, so blood, since it's connective tissue, has cells, and you know what those are: red blood cells and white blood cells. And it has matrix. And the matrix is ground substance. And I'll get to the components of that. But these are some of the components. So the bold print term here is components. What's the components of blood? One, formed elements. And formed elements means cells. The next thing, plasma. Plasma is the matrix. $55% of blood in this jar is plasma. The other 45% is the formed elements, the cells. Okay? So what I'm going to do is we're going to take plasma first, and then we'll come back to the formed elements. Okay, what is in plasma? 92% of plasma is water. 92% of plasma is water. 7% is proteins. And the major proteins I've just outlined right there. Albumin, white of an egg, of a chicken egg is albumin. Globulin, that's where we get the proteins for hemoglobin. That's where the base comes from. The next two, fibrinogen and prothrombin, are used in helping form this blood clot. These are in a neutral, or essentially a hibernation state. So they're going to be triggered by something to become something else to start a blood clot. And that'll be the last thing we talk about in this series. So we know that 55% of blood, you got a handful of blood in your hand, 55% of it is plasma. Of that plasma, 92% is water. 7% are proteins, and these are just a couple of them. And that leaves 1% inorganic, all other things. I'll just put other. Good? Okay, I'm going to erase the board, and we're going to put some more stuff on here and continue. Break. How's it? Okay, we left off and we were talking about the components of blood. And we talked about blood being 55% plasma, 45% formed elements. We talked about the major components of plasma. We know that 92% of plasma is water, 7% is protein. And I gave you a list of 4 or 5 common proteins that are circulating in the blood. 1% is all the other stuff, the calcium, the sodium, the chloride, all the other things that are in your blood makes up that 1%. Now the next step is the formed elements. You have looked at the formed elements twice already this semester. Once under the microscope, when we looked at, in the microscope lab, we looked at blood as connective tissue. And we saw that most of it is those pink little donuts out there, which are red blood cells, properly called erythrocytes. And then we saw some little bit larger stuff that had a nucleus in it the nucleus was usually kind of purple. Those were the white blood cells, of which there are 5 types. So we've talked about blood before. We've seen it under the microscope. And now we're going to go into detail. In formed elements, there's 3 types of cells: red blood cells, white blood cells, and platelets. Written right here. Which are properly called thrombocytes. Okay, red blood cells. Red blood cells are properly called erythrocytes. We talked about the process of making them, the red blood cells, was erythropoiesis. Hemopoiesis is just making of blood. Erythro is specifically red blood cells. And that happens in spongy bone, red bone marrow, myeloid tissue. We talked about that already. And you saw the bone that was in lab where you could actually touch that. Okay, characteristics of red blood cells. Number one, they're biconcave. Let's see what the heck that means. I'm going to draw three shapes up here. And we're going to label these shapes A, B, and C. B is the shape of a red blood cell. Question is, which one has the most surface area, A, B, or C? This is the shape of a red blood cell. Think about this. Embedded in the phospholipid bilayer, remember what that was, is an enzyme called hemoglobin. Hemoglobin transports oxygen. That's all it does. So when we talk about a red blood cell, the red blood cell doesn't do anything. It's what's embedded in the phospholipid bilayer, which is hemoglobin, attaches to oxygen, and that transports it to the cell. So that means with every red blood cell, you want the most surface area as you can possibly get. We've been talking about surface area throughout the semester. One with the mitochondria, the matrix, and the crista. We also talked about the villi in the intestine to increase surface area. And what increases the surface area in red blood cells is the shape of B, okay? But it comes at a sacrifice. A and C can have a nucleus. B cannot, because it won't fit in there. And you have to have a nucleus in order for the cell to undergo protein synthesis, cellular respiration, and reproduce itself. So that means red blood cells cannot reproduce themselves because you had to give up the nucleus. So my question to you is, why not take C? C has the most surface area, just like B. A, you lose surface area. So you want to become most efficient. Every time a red blood cell leaves the lungs, you want the maximum amount of oxygen attached to it. You can't get that with A. B, you have to give up your nucleus, so it can't reproduce itself, which means it has to go to a stem cell, which is in the myeloid tissue. C would handle everything. You increase the surface area, reproduce itself. But then the cell is even larger than A, which means your blood vessels would have to be bigger, your capillaries would have to be bigger, your heart would have to be bigger, and you'd have to pump more blood for each heartbeat. So this one is out. But if you look carefully, you'll see that B and C have the same surface area. So if you take instead of convex, you concave it, the surface area is the same between B and C. The body is elected to go with B, even though you have to reproduce it someplace else, because then everything can be smaller. So you can get, picture a beaker full of blood with C. It would be overflowing, but in B, it would be at the brim. So we have biconcave, and now you understand the reason for it, to have more blood cells per milliliter. Okay, you also heard the term hemoglobin. Okay, we already talked about globin in the blood. Globin is the protein, heme is the cofactor, and this is iron. So iron plus protein, this particular protein forms hemoglobin. Hemoglobin is an enzyme that attaches to oxygen so it can be transported in the vascular system to the cell. Now, remember we were talking about that volume of blood. Okay, if this was a container of blood and I put it in a centrifuge and I spun it, what a centrifuge does is separates things out by weight. Blood cells are heavier than white blood cells. If we spun blood, we would get So let me do it down here. We take blood and we spin it, centrifuge. We will get two components, formed elements and plasma. We said plasma is 55% of what's in that. And when we spin it with, we spin it in like a test tube, and it separates out. And the formed elements is 45%. We've talked about that 3 times now. We already talked about the plasma. Now we're going to spin the formed elements. In other words, we're going to separate the cells. Okay? When we do that, we're going to get red blood cells on the bottom because they're the heaviest. Then we're going to get a combination of white blood cells and platelets because they're the lightest. When we spin the blood and separate out the red from the white, the red is called hematocrit. Hematocrit is only RBCs. The remaining part is called a Buffy coat, which is the white blood cells plus the platelets. Good? Okay. Let's talk a little bit more about the red before we go into the white. When we have some terminology. A red blood cell, a hemoglobin molecule that has oxygen attached to it is called oxyhemoglobin. So in this red blood cell, in the phospholipid bilayer that we're talking about, is hemoglobin. When hemoglobin has oxygen attached to it, this takes on a new name. It's called oxyhemoglobin. Still a red blood cell, but that's where you get the oxy. Now, we were probably taught a long time ago that you breathe in oxygen, but you're actually breathing in air. So when you breathe in air in your lungs, it separates out oxygen from the other gases, nitrogen and stuff like that. Can you hold it? Okay, where we left off was, as I said, that we breathe in oxygen. We don't breathe in oxygen, we breathe in air. And air in the air is partly oxygen. We separate that in the lungs. The lungs diffuses into the blood through capillary beds. We're good with that. What you were probably taught, or somewhere along the line you felt this to be true, is you breathe in oxygen, it takes it to the cell offloads carbon dioxide, which is somehow oxygen was turned into carbon dioxide, and then you bring it back to the lungs on a red blood cell and you offload the carbon dioxide. That's yes, no. The yes part is 20% of carbon dioxide is carried in the blood from the cell on a hemoglobin molecule, only 20%. Down here. So 20% of carbon dioxide, wrong part. 20% of carbon dioxide that's transported in the blood is transported on a hemoglobin molecule. And when oxygen was attached to hemoglobin, we called it oxyhemoglobin. When carbon dioxide is attached to it, it's called carbaminohemoglobin, 20%. Okay, now we're dealing with 100% carbon dioxide. 10% is carbon dioxide gas diffused into the blood. So that lets us know where the 70% comes from. This ion that I'm going to come back to later, called bicarbonate ion, 70% of CO2 is transported in the blood on a carbonic acid. Let me take that again. A bicarbonate ion forming carbonic acid, which is what I have here. Okay, so back over here, carbaminohemoglobin, when you see that, that's hemoglobin with carbon dioxide attached to it, 20%. Okay? Bicarbonate ions, which we're going to find out later are called buffers, can absorb the majority of it. And we said that was 70%. And when bicarbonate ion has carbon dioxide attached to it, it forms a compound called carbonic acid. Okay. Next. We said that blood cells are made in myeloid tissue, commonly called red bone marrow, which we know that is spongy bone, which is spongy bone. The life expectancy of a red blood cell is about 120 days. So that means red blood cells are constantly being broken down and then reborn. The major breakdown point for red blood cells is the liver. And the process of breaking it down, as we remember, is called phagocytosis of anything to eat cells. And this cell, we're talking about a red cell, red blood cell. That means we're going to have components coming out of it. So the liver is going to be breaking this stuff down because the cell is all beat up, and we're going to take the components and we're going to do something with them. Sometimes we have to excrete them in urine or in feces. The other thing we can possibly recycle. The biggest thing is is bilirubin. And the heme portion of let's back up. Ready? Okay. The breakdown of any cell is called phagocytosis. When we break down a red blood cell, one of the major components is hemoglobin, obviously, because that's what's in the phospholipid bilayer. The breakdown of the iron results in a compound called bilirubin. Okay? Bilirubin is excreted in urine and in feces. Another component is iron with the phospholipid bilayer breakdown, and that forms bile. And what the body does is it uses bile to emulsify fats. It takes the globin, which we know is amino acids, which is protein, and recycles that. Now, we've heard of the term anemia before. And probably all of us have felt anemia is a lack of iron. And that is yes, no. We have two major types of anemia. Let me put it this way. Anemia is low functioning red blood cells. So how can you get low functioning red blood cells? Lack of red blood cells, lack of the protein globin to make hemoglobin, and lack of iron. The lack of iron is iron deficiency anemia. The lack of amino acids forming the proteins is called pernicious anemia. If you have a patient that is bleeding, they're becoming anemic because they have a low red blood cell count, which is the definition of anemia, functioning red blood cell. Okay, let's go from red blood cells to white blood cells. There's two groups. White blood cells, there are 5 types. There are 5 types. And they're grouped arbitrarily into 2 groups. Non-granular and granular. Over here, I have them lined up. Now remember, there are 5. First, we've already talked about a red blood cell, properly called an erythrocyte. The 5 white blood cells are called leukocytes, grouped into 2 groups. Non-granular, no granules, and the granulars. The granulars end in the suffix "phil." The non-granulars end in the suffix "cyte." So this is a monocyte, this is a lymphocyte, this is a neutrophil, this is a basophil, and this is an eosinophil. Now, you remember when we did tissues, I had this hanging on the wall. And this showed the 5 types of white blood cells and a red blood cell and what their generalized function was. The first one And the last one are the cytes. The other three are the fils. The lymphocyte, remember I gave you the mnemonics, never let monkeys eat bananas. Never for the neutrophil, let lymphocyte, monkeys, monocyte, eosinophil, eat basophil, bananas. That's the order of frequency in the body. So the most common is a neutrophil. It does its job by eating. Phagocytosis, we just talked about that. So when you get something in your body that doesn't belong there, a bacterium, a virus, or whatever, this is going to be your first attack blood cell, which is your immune system. The second one, and the most common one that we hear about, is a lymphocyte. And it works by antigen-antibody complex. Now, I'm going to go over to here. Here's what I'm talking about. An antigen is something in your body that doesn't belong there, like the coronavirus. Antibody is something that's produced by that lymphocyte in its genetic code. It is a protein. And that protein attaches to the antigen and signals either a monocyte or a neutrophil to come and eat it, or it's picked up by the kidneys and excreted. So it essentially, antibodies neutralize antigens. Example of the coronavirus is an antigen. And what they're trying to do with a vaccine is to create antibodies before you get the antigen in there, in your body, so you can attack it. Okay, shifting gears, back over here. Two groups. Non-granular, which are the lymphocytes, which are the second most common, and monocyte, which is the third. Unique about monocytes, we've already talked about it. We talked about a monocyte. It's circulating in the body in a dormant state. But remember, when histamine is released from a basophil, we had diapedesis. Remember that? And diapedesis was continuous capillaries becoming discontinuous. So there's gaps in the capillaries. When you have gaps in the capillaries, this guy can come out of the circulatory system and get around your cells to police the antigens. When it does, it's activated. When it's activated, it's no longer called a monocyte, it's called a macrophage. Just like it sounds. Phage to eat, macro, it's a big eater. Okay, so once again, two groups, non-granular, sometimes it's called agranular, and then granular. The non-granular, lymphocyte, monocyte, I told you how those work. The next one, granular, those are the fils. The most common white blood cell in the body in a normal human is a neutrophil, and it's phagocytic. Eosinophil, that's the fourth least common, and it's used for parasitic things to get in the body. So if you have a parasitic infection, your eosinophils will go up. Basophil. Basophil is a white blood cell that's circulating in your blood to monitor normality and antigen attack. If you have an antigen, which can be pollen, basophils secrete histamine. And under the influence of histamine, capillaries become discontinuous, monocytes become macrophages. So they tie into one another. Now, some terms. Leukopenia means low white blood cell count. It's just a term. Low white blood cell count. High white blood cell count. Leukocytosis. That means you have an abnormal white blood cell count for whatever reason. It's just term, describes that. I want you to see polycythemia. That means higher than normal red blood cell count. Leukemia. That's cancer of the blood, white blood cells. Remember we talked about that? The general term is neoplasia, leukemia. All right. Next one. Let's go to a blood clot. Keep it running. Let's go down here. When our closed circulatory system becomes open, the body, if it's not too big of an opening, can close it off using what we have on board already. That's a blood clot. It's going to require a couple of enzymes and our platelets, which are properly called thrombocytes. And here's where I'm at. Blood clot. Okay, circulating in your blood right now, you have two proteins, enzymes. One, prothrombin, two, fibrinogen. We talked about that when we talked about some of the common proteins that were in plasma. Okay? They're in a sleep state. They're hibernating. So let's say this is a wall of a blood vessel. This is a nucleus. We know it's made up of cells, just like everything else. If one of these blood cells is lacerated, inside the cytoplasm, remember cytoplasm, cytosol, cytoplasm, is an enzyme, and this is not a scientific name, but it is. It's called factor X. Factor X causes, so if I'll put factor X plus prothrombin equals thrombin. Thrombin, when it comes into contact with fibrinogen, gives us fibrin. Fibrin is what we want. That's how we're going to plug up that hole. This blood clot, the wires represent fibrin. The little blue circles represent platelets or thrombocytes. So I want you to imagine you're washing your hair and you're shampooing it. And you know when you're rinsing it, you look down in the drain, and some of the hairs have started to plug up the drain, and the tubs or the tub that you're standing in starts to fill up with water. Very, very similar to forming a blood clot. The hair is fibrin, long proteins. So let's say this is the interior of a blood vessel. I'll just put inside. Now, I'll make the fibrin black. So here's a fibrin. Here's another fibrin. Here's one that got out. Here's another one that got out. Another one got out. Another one stopped. Another one stopped. Now, the more that stop, the more we start to plug up the drain, just like we did with the hair in the shower. And pretty soon, it gets pretty plugged up. But there's little tiny holes that still allow the seepage of blood. And I'll do red for the thrombocytes. Then the thrombocytes fill it up so it's an actual plug, nothing more that can get out. This, on the surface of our skin, we call a scab. When we can't see it underneath it, this is what it is. This is a blood clot. What I drew right there in the black and red is this right here. Made up of thrombocytes, which I did in red, and fibrin, which I did in black. a scab on the inside of a blood vessel, because that's where the blood's coming from, is called a thrombosis. So picture it as a scab on your skin, a scab inside your blood vessel, called a thrombosis. This is a thrombosis. Okay? Now, you know, after surgery, you've got a lot of thromboses because many blood vessels have been cut. So you have clots all over. So right after surgery, they tell you do not move until new cells have undergone mitosis. We patched up the holes, and then enzymes will break down that thrombosis. If, and it happens, that thrombosis breaks free and begins circulating in the blood, it's no longer called a thrombosis, it's called an embolism. You've heard of pulmonary embolism. Embolism just means a moving blood clot. Okay, to finish up with a couple extra more terms. Heparin. What the heck does heparin do? We talked about histamine. We know histamine causes diapedesis, remember? Heparin is also released by a basophil. And what it does is it inhibits factor X. So heparin neutralizes factor X. Therefore, prothrombin can never become thrombin, which causes fibrinogen to become fibrin, there you leak. Now, you've heard of leeches, more commonly mosquitoes. They're surviving on blood. So what they do is when they find a host, they will cause a wound to occur. So they'll break the skin, get to a blood vessel, then secrete heparin so you constantly bleed. And then they lap up the blood. Do you understand the function of heparin? Okay, next two terms, acidosis, alkalosis. Blood has a pH of about 7.3, 7.4, a pH. We know that a pH scale is 0 to 14, with 7 being in the middle. So blood is just slightly basic. Another word for base is alkaline. If your blood starts to go back down towards the 5s and 6s, the condition is called acidosis. Okay? If it goes above 7.4 into the 8s, the term for it is alkalosis. Now, question. We realize that in our day-to-day living, we're breathing air that is not 7.4 or 7.3. We're eating foods and drinking things that are not 7.4, 7.3, which means we're subjecting our body to either an acid or a base environment. How can we do that? And the answer is we have buffers in our blood also. We talked about it before. The major buffer is bicarbonate ion. And what bicarbonate ion does, it can capture and hold on to hydrogen, which is acidic, which makes things acidic. Free hydrogen makes solutions acidic. And OH, hydroxide, makes solutions basic or alkaline. So when we take a food that has extra H's on it, means it's an acid, bicarbonate can come over and grab the H's and neutralize it back to 7. If we have things that make our blood higher in content of OH's, hydroxyl ions, then the buffer bicarbonate ion can grab a hold of that and neutralize it. We're back to 7. Question is, can I run out of buffers? Yes, I do. If I run out of buffers, what's going to happen to my blood? It's either going to become acidic, depending on if it's alkaline or acidic. It's going to become acidic, acidosis, or it's going to become alkalosis. That can happen. When that happens, it starts changing the three-dimensional structure of proteins. As you remember, three-dimensional structure of proteins can be altered by two things, temperature and pH. Proteins have to have a certain three-dimensional structure in order to do their job, which is either catabolism or anabolism of compounds in our body. If you have that protein that normally does that and it can't do it because its shape has been changed, then you end up becoming ill or sick or even dying from that. Okay. A couple more terms before we finish up. Blood types. What are the blood types? You've probably heard A, B. Now you want to throw in O. Essentially, I'm going to put O down here right now. You have blood types A and B. The way you get the term A-type blood is you have an antigen. Remember, I was talking about the antigen? That as you're developing in the uterus, the body is understanding the antigen and it neutralizes it. So type A blood people have the type A antigen, but it doesn't affect them. The same thing for B. B has the B antigen. Type AB has both A and B antigen. O lacks antigen. So if you were to take a person that had B-type blood, which means it has an antigen, you put it in an A-type person, you're going to get coagulation because you have no antibodies for it. If you put type O blood, A or B or AB into O, they're going to coagulate because they don't have any antigen. Okay? So the way you got type A blood, type B blood, type AB blood is signifying what type of antigen you were born with or at conception, development. Okay? So let's take a look at that. So the universal donor of blood, what do you think? It's going to be O. Because you can give O to A. Will it do anything? Nope, because they don't have any antigen. What about B? Nope, because they don't have any antigen. What about AB? Nope, because it doesn't have any antigen. So O is a universal donor, and that's why everybody that has type O blood, they're screaming for your blood. Now, what is the universal recipient? Recipient means somebody that I can give blood to. Well, it's AB, because AB can get blood from A because it has the antigen. It has the antibodies, sorry. B, because it has the B antibodies. And it can get it from O because O doesn't have any antibodies whatsoever. So the term for that is universal recipient, which is AB. Universal donor, O. We talked about a buffer, and the main buffer in the body is bicarbonate ion. We said that when it transports carbon dioxide, it forms a compound called carbonic acid. We also did the 70, 20, 10. Bicarbonate ion transports 70% of carbon dioxide. Carbon dioxide gas diffuses in the blood, about 10%, and Carbaminohemoglobin, 20%. Good? Now, go over each one of these terms, make sure you're very comfortable with each term because every one of them is showing up on your exam, either as a question or as an answer or a combination thereof.